This exercise continues with our section on food microbiology, and specifically it's talking about wine fermentation processes. Wine, most people are familiar with, is made from grapes, and traditionally it is made from grapes. You get the grapes, collect the juice basically, and ferment it to make the wine. You can actually use any type of fruit, though, to make wine. So a lot of people experiment with apricots or peaches, etc., and making other fruit wines, but traditionally you use grapes. Excuse me. To make white wine, you use white grapes, or you can use red grapes, but you have to remove the skin in one of the very first steps after harvesting the grapes before you crush it, so that the red pigmentation from the skin does not get ultimately into the wine. Red wine uses red or purple skin grapes, depending on which specific type of wine you're making and what is the taste or the flavor of the wine that you're trying to get. White wine typically is fermented longer than red wines as well. So the steps of making the wine, whether it's red wine or white wine, first is a crushing stage, then you're going to ferment it, store it, filter it, and then finally age it. And so this slide shows the steps of just basic winemaking. So obviously first you have to collect your grapes. It's a very scientific process now they have of what areas and which location is best for growing which specific type of grapes to get the type of wine that you want. And in this first step here, before you crush them, remember, for white wine, you're going to be removing the skins. So you collect the grapes, remove the skin if you're making white wine, and then you're going to crush them. And then basically the liquid, the resulting from the crushing, you collect that. You're going to add sulfite to the solution. This is to kill any of the microorganisms. There could be bacteria there. There could be yeast on it that you're not really wanting. So you want to kill first the microorganisms that you don't want there. So that's why sulfite is added. After that, you add the yeast that you want. You're going to use yeast to help ferment the wine. So that's the step now where you're adding the yeast that you want, and then you mix it in and you let it ferment. So the yeast is breaking down those sugars and making the alcohol, the wine. After the fermentation process, you collect the fluid, and it's going to usually be pressed. You collect that fluid, and then it goes through a clarification process. This is usually putting it through vats. The clarification process is removing large solid materials from the solution from the wine. Then it's going to be filtered. After filtration, it's going to be aged. It's usually placed in barrels for this aging process. And how long is it aged? Once again, it depends on what type of wine you're trying to make. After the aging process, then it is bottled. Typically, the older the wine is, the darker the color. So like a red wine, if it's fairly what they call young, fairly new, it's a lighter red. A deep red is often associated with an older, longer aged wine. And same thing with the white wine. It's usually a very light or clear almost color, but it may be more of a yellowish color for ones that are aged and are older. It just depends on which flavor you're trying to get for it. Now with the wine fermentation, obviously this is a specific field or subset within food microbiology, has been extensively studied. People, with their vineyards are always kind of experimenting with different types of grapes, different type of yeast. And the location is actually very critical for the growth of the grapes. And just trying to get different flavors and all that. Now, With fermentation, you are only going to get a certain level of alcohol concentration. There's a limit as to how high. It's not like, oh, I can let it ferment longer and it's going to be a higher alcohol concentration. No, there's the limit. And so to increase the alcohol concentration, sometimes what people will do is then you can distill the wine or whatever product you're making. Distilling is going to give you a higher alcohol concentration. So to make some of your other alcohol beverages, some of your other liquors, you use different starting materials. So instead of grapes, like if you take sugar cane and you run that through the distillation process, that is how you make rum. So rum is coming from sugar cane. Vodka, they use potatoes. Whiskey, you use various types of grains. And to make brandy, they actually, the three most popular types of brandy, they're either using grape, peach, or apricot wine. So they're taking the wine that's already been fermented and then they distill that. Now, in our lab, we do a much simpler method of this. We simply take flasks that has, we get some type of a fruit juice. It may be apple juice. Oftentimes we try to get grape juice or cranberry juice. So we just pick some type of a fruit juice. And we do add just regular yeast, like if you bake bread. It's Saccharomyces cerevisiae is the genus and species of it. But it's just like the regular yeast packets that you buy in the store. You add some of the yeast to the flask, and then we just let it sit at room temperature. Now in the flask, you notice we put a balloon over the top. These balloons, when we first started, were not inflated at all. As the yeast is fermenting the sugars in the juice, it's producing that alcohol, and one of the side products is gas, typically CO2. And so what happens to that gas that's being produced? That's what's inflating the balloons. And so that's the way we just sort of monitor. It's a simple little exercise, easy to do, just to see the fermentation. Now, unfortunately for the students every semester, yes, I have to remind them this is done in a lab and you cannot consume any food or drink in the lab. So no, at the end of this, in the lab, you do not get to taste test. We don't have any taste testing labs for this. But it's a quick, easy way of showing, the very basic process of the wine fermentation. And even in these flasks, afterwards, students will usually look at it and you can see sediment that has settled down on the bottom of the flask. And so that shows why you need to have the clarification process to remove the larger substances, followed by filtration to remove any remaining, especially smaller particles in there.